Hello, everyone, and welcome to today's live broadcast, Investigating Colorectal Cancer Using Epithelial Organoid Cultures, presented by Dr. Kretzschmar, Postdoctoral Researcher, Hubrecht Institute. I'm Alexis Krauss of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Thermo Fisher Scientific. For more information on our sponsor, please visit thermofisher.com. Now, let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question and click on the Send button. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, click on the arrows at the top right-hand corner of the slide window. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Answer a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I'd like to now introduce our presenter, Dr. Kretzschmar. Kretzschmar received his PhD from the University of Cambridge in 2014. During his doctoral training under the supervision of Fiona Watt, he studied the role of canonical WNT signaling and the plasticity of adult murine skin and epidermal stem cell heterogeneity. Subsequently, Kai joined the laboratory of Hans Clevers at the Hubrecht Institute in Utrecht to investigate the heterogeneity of the Tumour microenvironment in colorectal cancer. Dr. Kretzschmar, you may now begin your presentation.
Thank you very much for this kind introduction. The title of my presentation you already heard. I will talk about using epithelial organoid cultures to investigate colorectal cancer. I have an overview provided. I will first actually introduce you to colorectal cancer, give you some facts about it and some kind of basic biology to this disease. And then I will introduce actually what epithelial organoid cultures are and how we can use them to grow normal colon epithelium as well as colorectal cancer tissue. I will continue in the third part of my talk showing you some data that we have published. How we can actually use organoid cultures to biobank actual cancer tissue from patients and use them for drug screens, analysis, etc. In the last part of my talk, I will actually show you how we can use normal colon organoids and genetically manipulate these to study and model the progression of tumors as you would see them in a patient. Okay, let me start with an introduction to colorectal cancer. Colorectal cancer is actually one of the most common cancers in the world. It's in the top 5 of new cancer cases per year, but also actually in the top 5 of cancer-related deaths per year, and therefore, of course, a very important cancer to be studied. Classically, it's been believed that colorectal cancer arises in a canonical way from normal colon epithelium, which acquires mutations that lead to the development of a small adenoma, which then progresses into a large adenoma, and then eventually, in very aged patients, it presents itself as a proper colorectal cancer carcinoma. And a study by Augustine showed actually that these steps towards the disease are associated with mutations in very critical genes such as APC, KRAS, P53, and SMAD4. So the mutations that you find in APC and KRAS actually activate the pathway, so they're chronically upregulated and active, whereas P53 and SMAD4 are usually mutations that inactivate these oncogenes and therefore allow progression towards the cancer. Now, this classical focal-gram colorectal cancer is actually only one of 4 subtypes that are nowadays studied. There is another characteristic one, which is called microsatellite unstable tumor, which is the one you can see on the left. Then the next type of colorectal cancer is the so-called canonical one, that I just presented. And then there are two other types, a metabolic one and a mesenchymal one. And all these four types can actually be studied in our epithelial culture system. Now, why is it important to have a good in vitro method, and what in vitro methods are there to study cancer? Well, of course, you cannot study cancer very well in a patient, so you need to use some methods outside the human body. And traditionally, people use cancer cell lines that are derived from tumor biopsies, but these have several issues. For example, of course, they don't show the heterogeneity within a tumor, so they tend to grow very clonally, and at some point will be representing essentially one cell found in a tumor. And they're also genetically very unstable, acquire a lot of mutations due to the culturing. The other system that people have used is transplanting tumor cell lines or tumor cells from the patients into mice. And that, of course, has issues because humans and mice are not alike. You need to use immunodeficient mice and therefore cannot study how the immune system, for example, influences tumor growth. And similarly to the colorectal cancer cell lines, you do not have the heterogeneity of the tumor reflected in the system. And last but not least, this system is very low in throughput and very expensive. So the development of organoids, and I'm showing you in the bottom essentially our system, how we actually generate these. We take a biopsy of the adult small intestine of a patient, for example, or of a mouse, and also the colon, of course. We can isolate the crypts, which contain the stem cells, and we can from there isolate the stem cells and put them in an extracellular matrix-rich environment and grow them up into so-called intestinal epithelial organoids. There are other ways to generate organoids that I will just show. One of them is actually directly taking the crypts and putting them into the martigel or the BME, and you can generate the same organoids. And there are two other ways you can generate organoids, but these are not epithelial organoids because they also contain the mesenchymal cells surrounding. So the rest of my talk will be concentrating only on the cells, on the organoid cultures I showed you in the beginning, which we call epithelial organoids. Now, if you take a normal intestinal stem cell, LGR5 positive, you can see now on the screen how this actually develops over time in culture. So you see from a single cell over about the 2 weeks, the whole cell will start dividing, will grow, will generate more and more cells, and will essentially generate an epithelial organoid as we have them in culture. So this is actually what happens there. It's a very defined culture medium as well that we have associated with it. So what are the characteristics of these organoids? They are three-dimensional, and as I said, they can be established from a single adult stem cell, and they require certain niche factors that are important, such as EGF, noggin, and arspondin. But they are all epithelium. There's no mesenchyme, no cells, no fibroblasts in there. And they, in the beginning, of course, are generated from a stem cell, but as time progresses, the culture progresses, these organoids will also contain the differentiated cell types found in the intestine, for example, enterocytes, and they can be expanded indefinitely, and in contrast to the other methods I introduced to you earlier, they're genetically very stable and actually only acquire mutations at the similar rate as you would find in the human body. Now, we thought to use this epithelial organoid culture system and use this to actually maintain colorectal cancer organ cells as well. And this is what you can see here. So we essentially take the tumor resected tissue, so usually a part of the colon is taken out, and take a biopsy from the tumor and take a biopsy from the about 10 to 15 centimeter distant normal epithelium, make single cell preps or isolate the crypts from the normal colon, and then can put them into BME. And as you can see on the screen, we can generate these organoids as I showed you earlier. So this is our starting point for the continuing studies. Now you could ask, how can we make sure that in the tumor, for example, we do not have any normal tissue left? Well, that's very easy, because actually a large fraction of the tumors have mutations in the Wnt pathway, almost 100%. So they have actually a chronically activated Wnt pathway, and therefore do not require a condition medium that we use to maintain the normal colon organoids. that is a wind condition medium. So essentially, by growing the colorectal cancer organoids in the absence of this wind condition medium, we can ensure that we only grow tumor tissue. And in the normal tissue, we usually can actually identify it by sequencing whether it's normal or not. Usually the growth will tell you as well whether it's normal tissue. Now, we can use these colorectal cancer organoids and the adjacent normal tissue to generate a biobank by essentially taking a lot of biopsies from patients that gave informed consent. And we have created a foundation that actually takes care of biobanking this. And then you can see on the screen several other tissues and associated cancers for which we have established living cancer organoid biobanks. And what do we do with this? We can use these organoids and these biobanks as an experimental tool where we can ask basic biological questions related to stem cell biology, infectious diseases, or cancer. And indeed, we can also use these to test drug efficiency, do genomic analysis, predict what drug might be used. So for diagnostic purposes, organoids can be used as well. And lastly, this is something that will still take some time until it is possible, although proof of principle experiments have been done already. These biobanks, these organoid biobanks, can be used for therapeutic use, for example, in the sense of transplanting organoids grown up to organ size or near organ size, or at least to contribute to parts of the organ. And we can use them also for gene correction, transplanting gene-corrected tissue back into a patient. But this is still something that we need to improve. Now, I will introduce you to our colorectal cancer organoid biobank that we have created 3 years ago, which actually is a biobank of 19 patients and their organoids grown from their tumors. And we have used this in various ways. We analyzed the DNA sequence so we could identify the tumor mutations present. We studied the transcriptome of these organoids and then performed drug screens and other things to see whether we can actually use the gene expression data to predict if particular drugs work on the tumor. And this we can always do in comparison to the normal colon organoids, because we have those also in culture. Now, if you don't believe me, I will show you some slides showing actually that the organoids that we grow from the tumor epithelium very much reflect the epithelium in the original biopsy. So you can see some hematoxylin and eosin stainings that show that the way the tumor grows as organoid is very much alike to what you see in the patient. So the histological analysis suggests this. So if you show this to a pathologist, they really recognize the resemblance of the organoids and resemblance towards the patient's original tumor. What we've done in addition is we looked into how similar the genetic alterations, the mutations are that we find in the tumor, and then in comparison to the organoids. And we could see that the overlap between the two, between the original biopsy and the organoid, is very striking. It's close to 90% in many cases. So showing that the organoids really truly reflect what you find in the tumor epithelium in the patient. And of course, we also find the typical mutations that I introduced you in the beginning. APC on the top right, P53, KRAS, are typical mutations found in colorectal cancer, and they are also present in our organoid biobank and also in organoids collected or generated from patient samples collected a bit later. Now, these tumor types that I mentioned in the beginning can be identified by doing transcriptional analysis. That's what we did also with the organoids. And I'm not so sure if you can see this on the screen, but if you look in the top bar, you can see these orange dots, and they actually represent the organoids that we have generated and their gene expression. And you can see they fall into all the 4 types of colon cancer, so they really represent the whole spectrum of colorectal cancer. Now, in addition, based on, for example, the gene expression pattern and the DNA sequencing, we can make predictions whether a particular drug that targets a particular signaling pathway will work on patients. And you can see on the top, Nutlin 3A is a drug that actually will act on cells that have still normal p53, and you can see that there the IC50 of those cells that still have a normal wild-type p53 is much, much lower than in those that actually have a mutation in p53, suggesting that a drug that targets this pathway will, of course, only work best in patients that still have wild-type p53, and those that have a mutation in this pathway will probably not have a therapeutic benefit. The same is true for cetuximab, which acts on the EGF KRAS pathway. You can see that those that have a wild-type KRAS still show a good response, whereas those that have a mutated KRAS are not sensitive to this drug anymore. So if we can just generate these organoids and then actually look in their gene expression pattern, we might be already able to predict whether a particular drug used on a patient will actually work or not. Now, culture conditions are very critical, and this is what you can see on the next slide, that the normal epithelium is very much dependent on all these key niche factors such as Wnt and R-spondin, EGF, p38, noggin, and TGF-beta, whereas the tumors are more or less actually able to grow independent of Wnt and R-spondin, for example, but also other components, because they simply have mutations in the KRAS pathway, therefore they will not require EGF and other signaling pathways, and therefore will not require any of the other drugs. Now, I will summarize this by playing a video that will follow.
Organoids are long-term cultures derived from diseased or healthy tissue of patients or animals. This can be done for almost all patients and for virtually all organs, and with regard to different diseases such as hereditary disorders and cancer. Both healthy and diseased tissue is isolated from an organ. These small tissue fragments are placed in culture. The isolated stem cells then grow into organoids. These organoids are very similar to real organs and form a reliable testing environment for medicines against all kinds of diseases. Using DNA sequencing, errors in the genetic code can be detected. We can also screen multiple combinations of medicines selectively and reliably on a large scale. Organoid technology is a powerful method which can be used to design and test patient-specific medicines.
Okay, in the last part of my presentation, I will introduce you to ways that we have developed to actually start from normal colon epithelium and generate organoids from this, and then introduce mutations that are relevant for cancer progression, and then see how things change in the organoid. And I introduced you in the beginning already these 4 key mutations that are found in the classical colorectal cancer: APC mutations, KRAS, p53, and SMAD4. We can actually replicate this by starting from normal tissue and then see what happens. We use CRISPR-Cas9 for this, which is a genome editing technique that allows us to directly introduce mutations in particular genes. So we can generate so-called guide RNAs that actually target these, and then what we used is we modulated the composition of the medium that I showed you earlier, and thereby select for only, for example, organoids that can grow in the absence of EGF in the medium. Those organoids that have a KRAS mutation, for example. I will give you an example on this using the canonical Wnt signaling pathway. That actually requires Wnt ligands to be active, and that's something you can see on the right-hand side. In the absence of the Wnt ligands, there is no gene expression of the target genes found, and therefore the target genes cannot be expressed. Proliferation is inhibited in those cells that do not have Wnt ligands bound to their receptors. In APC mutant organoids, though, this pathway is chronically active. Essentially, the Wnt ligand is not required for gene expression. So what you see on the left is actually present, so the Wnt ligand is not there, but actually, in terms of gene expression, the right-hand side is the case. So the beta-catenin modulator can actually bind to the target genes and express those and therefore stimulate proliferation. So essentially what we do is we take a wild-type organoid grown and add guide RNAs targeting the APC gene, and then we can wait a couple of days, and then we remove the Wnt condition medium and the respondent condition medium, which is also required to stimulate the Wnt pathway. And then after about 2 weeks, we can pick clones that grew in the absence of these two components and then sequence them and verify this. And this is actually what I show you in the next slide. So we can see that actually these organoids have frameshift mutations. So you can see this is how it looks. In the top left of the organoid panel, you see normal organoids growing in the complete medium, and all the other 5 boxes show you organoids that grow in the absence of Wnt and are respondent, and you can see only those that are actually mutant. So the bottom 4 pictures show you organoids that grow, and then the one that has guide RNA 4 grows best. We can confirm that these, and this is a representation of the mutation found in using guide RNA 4. There are frameshift mutations which essentially inactivate APC and allow the pathway to be chronically active. And we assess this using qPCR for axin 2, which is the target gene of this pathway, and you can see that even in the absence of Wnt and responding condition media, this target gene is still expressed very nicely, actually at higher levels than with it, really nicely showing that we have introduced these mutations. And now we can use this for all other mutations found, and this is what I show you here in this very busy slide. So APC knockout organoids were generated, KRAS activating organoids. They don't require EGF. Then in addition, organoids that actually had mutations in APC and P53, they were selected by the addition of nuclein and all the other combinations. Essentially, we generated organoids that contained up to 4 oncogenic mutations, and then we could study them. And what you can see is actually when we transplant these organoids, we can see that the quadruple mutant organoids that have mutations in APC, in P53, in KRAS and SMAD4 are the ones that actually generate a carcinoma when injected into mice. So therefore, very nicely replicating what you would see in patients. We can also see that those organoids that have all 4 mutations are chromosomally unstable and actually are also really, really perturbed and generate aneuploidy. What we did in addition, we transplanted these actually into the intestinal region and then studied whether they can generate metastases. And this is what we could, and also growth, and we could assess that those actually that have these 4 mutations present that are very much like a colorectal cancer grow very, very fast, significantly faster than all other mutations. And also in the next slide you can see the ones that have these 4 mutations are the ones that are actually able to metastasize. So we can actually really nicely replicate what you would see in a patient by genetically editing normal organoids. So starting from normal epithelium, we can really replicate what happens in a tumor in a patient, and a step by step, and really can see that only those that have all 4 critical mutations are actually able to metastasize. And there's a very low number of those that have SMAD4 wild type, so the step before the carcinoma, that are also able to metastasize. Possibly, and actually in this case, this metastasis actually was contained later on, as we found out, a mutation in SMAD4. So essentially it was a quadruple mutant organoid. Something else we can also do, of course, is we can also generate gene knockouts in the DNA repair complex to replicate organoids that are like microsatellite and stable tumors. And we can see that those that have a mutation in the MLHA1, which is a very critical DNA repair gene, start to accumulate a lot of mutations, and we can actually see that based on the organoids we generate, we see very, very similar mutations also in patient samples. So essentially we can replicate the mutations that you would find in a patient that has these microsatellite unstable cells, and we can find the same mutations by just artificially introducing these mutations, and we can therefore really study the tumor. So in summary, what we can generate is we can generate epithelial organoids from normal human colon and colorectal cancer tissue. From this, we can generate biobanks that we can use for drug screens and other things. And we can also genome edit these organoid cultures to study tumor progression, as well as study the origin of mutational signatures found in colorectal cancer and other tumors. I would like to thank the head of the lab, Hans Klevers, and also several other people that are still in the lab or have left the lab that have significantly contributed to the work. And I would like to thank all our collaborators in the Hubrecht Institute, in-house, in the University Medical Center, at the Netherlands Cancer Institute, as well as the Wellcome Trust Sanger Institute. And I would like to also thank all the funding agencies that make this work possible. Last but not least, thank you for your attention, and I'm looking forward to your questions.
Thank you, Dr. Kretzschmar, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just type them into the Ask a Question box and click on the Send button. We'll answer as many questions as we have time for at the end of the presentation. Questions we did not have time for today will be answered by Dr. Kretzschmar via email following the presentation. So let's get started. Our first question is, what is the success rate of establishing these organoids from patients?
Yeah, the success rate is actually, in the case of colorectal cancer, pretty high. It's 90–95% of the samples we receive actually we can generate organoids from. So it's in the case of this particular tumor type very, very good actually.
Our next question is, can single-cell mRNA sequencing be performed from the colorectal cancer organoids?
Yes, that works very well. So we've started, of course, with organoids from the mouse small intestine, and it gives very, very good results. And the same is actually also true for human normal colon organoids. You can get very, very good single cell transcriptome data, and actually for cultured colorectal cancer organoids, that is the same.
For how long can the colorectal cancer organoids be maintained in culture?
This is a very good point, actually. So we have some of these in culture for 2 years already, and they seem to be still going fine and still be very, very primary-like in the way they behave. So yes, so in principle, it can be a very, very long time, much longer than you can usually do with, for example, colorectal cancer cell lines that tend to kind of behave more like an immortalized cell line.
Are you primarily focused on the epithelial organoids, or do you culture with mesenchyme as well?
So in our system, we really specialize on epithelial organoids. So we only have the martigel that is supporting the growth, so we don't have an actual cellular mesenchymal niche. But in principle, that might be something that is interesting to look at. But for our purposes, in particular for the drug screens, we really like to see how the tumor epithelium itself behaves and works, and don't really do mesenchyme. So for our purposes, we do not do it, but I believe in principle it is probably possible.
What is the size of the organoids needed for the transplantation in animal models?
So I think there it really is dependent on what organoids you use and what colorectal cancer you actually implant, because I would say that in some cases you might have issues issues with the maintenance in the transplant. So maybe they, I don't know, they differentiate away or something like that, are not so stable necessarily. But a particular size, I actually don't know personally, because I'm not doing these kind of transplantations. But I would say probably organoids kept for about a week should be good. It's just a matter more of the number of organoids that you transplant.
How do you measure the viability percentage after drug treatment, whereas organoids are variable in size?
Yeah, this is indeed a very good point, and that's something that we in the beginning had really trouble with, and that is that we now actually filter the organoids so that they are maximum about 40 micrometer in size, and then really look at that to make sure that we do that. What we actually do to check for viability specifically is we use an ATP-based assay, which essentially gives you an indication of the overall growth, and then you need to, of course, have controls that have about the same growth efficiency, possible use the same organoid line as control to actually adjust for the differences in size and growth. But we usually tend to use organoids that are rather small because they can grow much larger than 40 micrometer actually.
Based on the morphology and growth rate when comparing normal organoid and CRC samples, we did not detect a very obvious difference. Do you think the in vitro culture system cannot translate the in vivo settings in some cases due to growth factors we add?
So I think the normal organoids that you saw in the presentation were actually cultured in medium that is more expansion medium, where we essentially maintain the LGR5 stem cells that really build the small intestinal or the colonic epithelium. So if you were to change some components in that medium, you would actually get them to differentiate, and they would look much different to the colorectal cancer organoids. I have to say though that the colorectal cancer organoids themselves tend to look really different depending on what type of organoids you have. And the ones that you saw in the presentation were actually those classical Fogelgram colorectal cancers, which have mutations in, for example, the Wnt pathway, and they tend to be very cystic. So you can also have other types, the mesenchymal one in particular, or actually also the microsatellite and stable one, where the organoid of the tumor is actually very dense, and you don't have this hollow cyst-like structure, and therefore you can really make a difference between the normal and the tumor. And let me just have a look. I think the critical point is that also in vivo, actually, there's a kind of a natural gradient of these growth factors, whereas in tissue culture, where we grow the organoids, that's something we cannot really replicate yet. So that would be something, if bioengineers would enable that somehow you could actually have a gradient of these growth factors implied in your culture system. You might actually have really clear differences between the tumor and the normal, because the tumor organoids are pretty much actually independent of these growth factors and probably could also grow in the absence of them.
Our next question is, how difficult will it be to derive organoids from other tumors without clear indication of the existence of cancer stem cells?
So I hope that the question means other tumor types that are not from the colon. So in most cases, the problem is really to find the best media composition for the organoids to grow. So you will have to start probably from normal epithelium maybe and try to get that to grow. And usually the tumor will grow in that condition too. But of course you will have to be sure that you somehow can select for the tumor, either by studying what mutations are in this tumor. I mentioned, for example, that you can use nutlin in case there is a p53 mutant, and this way you can select for cells that still have a wild-type p53 and would die in the presence of nutlin, and then only the tumor that is p53 mutant would grow out. So it depends. Generally, it should be possible if you really work out the right culture conditions. Cancer stem cells is another thing, because I think there is still a big debate whether cancer stem cells as such actually exist. For the colon, I can tell you that in most cases when we passage these organoids, we actually trypsinize them into single cells, and actually it's maybe not 100% outgrowth, but we get a very, very good outgrowth efficiency, 90%, 95%, which doesn't really make sense if you think that there are really dedicated stem cells in this tumor, if all of the cells that you have in the tumor seem to be growing out of virtually all cells. I should probably caution. So that's a different question, I think.
Is there a difference for the percentage of success in organoids culture from cancer patient samples and healthy patient samples?
So we don't get actually organoid samples from healthy patients. So of course, because what we actually, in our case at least, we get the colon epithelium from a section. So actually, the certain bits of the colon are really resected out, and from there we can take the normal epithelium. The only way you could have access to normal epithelial organoids from really a healthy patient is probably after a colonoscopy, where they take a biopsy, but usually that's not done if there is no evidence for any let's say indication of a disease, because of course you don't want to injure a normal healthy clear epithelium. So all the samples we have from human are actually from cancer patients.
Have you already established a biobank, and are any drug industries interested in cancer drug testing with organoids? Is it more efficient than cell culture?
Yeah, we have established a biobank. Actually, I mentioned this Hub biobank in the presentation. They actually have established, I think, more than 100 colorectal cancer organoid lines now. So that's already been done. And indeed, we have collaborations with the drug industry, teamed up with several companies, big companies and also smaller companies, and they use kind of these biobanks. So actually, also companies can approach the Hub biobank and ask for to buy organoid lines for their research, and it's going very well. It is indeed more efficient than, let's say, regular cell culture, because the colorectal cancer organoid lines really are reflecting heterogeneity of the tumor. They don't tend to be immortalized, so they stay rather primary, whereas the cell culture cell lines that you use, the classical colorectal cancer cell lines, they tend to be not behaving as they were when they were primary. So I would say it is pretty good. But what I have to caution is that organoid cultures in general is a very, very laborious and very money-intense work. So it would be, if you compare that, it really depends on your questions that you're addressing. It might still be better for you to use cell lines, because it's very, very, you need to put a lot of effort into this.
If you passage CRC organoids via single-cell dissociation, are you not worried to lose tumor heterogeneity and to select for high-growth cells?
So this is a very good question. That is surely possible. Although we have actually done single-cell sequencing from classical colorectal cancer organoids, and they tend to be not so heterogeneous as you would think. I think the heterogeneity between patients is much bigger. There are other subtypes, for example, I guess the microsatellite and stable ones, which seem to be much more diverse in their mutation status and in the mutations that they acquire. And that could be a problem, but with the classical Fogelgram colorectal cancer organoids, I would not be so worried. But of course, you can actually generate clonal cultures and then study as many clones as you like derived from the tumor, and then see how different they actually are. But that's of course a problem that is possible, but that problem is even worse when you study just regular cell culture 2D cell lines.
What are the special characteristics of the madrigal you use in your organoids? Is it commercially available? So
madrigal is generated by a company actually. There are several companies that produce something like Martigel, also something called basement membrane extract, BME, is actually very similar. That's what we mostly use actually nowadays. The special thing about this is that this is actually isolated from sarcoma grown on mice, and it contains extracellular matrix from this sarcoma, from this tumor, and stroma, and extracellular matrix proteins that really support the organoid growth. And that's really critical, so that the cells are in a 3D environment, not in a 2D environment, and that seems to be supporting their growth very, very well. So that's the special thing about it. Essentially, it's extracellular matrix.
Do you have experience with transplanting these organoids in zebrafish?
Short answer, no. And for us, really, we have not worked with zebrafish cells at all, in that sense that we try to generate organoids from them, and also, of course, have not transplanted them into zebrafish. So we only transplant into mice. That's the only in vivo experimental system we actually use.
Do you have biobanks of prostate organoids or only colorectal?
I believe the biobank does have also generated those from prostate organoids. Let me just go back to the slide so we can check together. Yes, it's listed there. So it is actually one of the tumor types where we do have biobanked organoids.
At what ratio can the colorectal cancer organoids be split?
They grow really, really well, and in most cases you can split them 1 in 6 up to 1 in 12. So you can generate a lot of cells in a rather short time.
All right, our next question is, how often do the colorectal cancer organoids have to be passaged or split?
Well, that passaging is really dependent on your question. If you want to propagate the organoids and keep them really nicely in culture, then this has to be done about once a week.
Thank you, Dr. Kretzschmar. Do you have any final comments for our audience?
Yeah, I would like to thank all of you for listening in, and if you have any questions, you can always get in touch with us at the Hubrecht Institute, and also get in touch, for example, with the biobank, the Hub Biobank, and see if there are any ways you can have access to these organoid lines. But overall, thank you very much for this opportunity and for your attention. Thank you.
I would like to once again thank Dr. Kai Kretzschmar for his presentation. I would also like to thank Labroots and Thermo Fisher Scientific for making today's educational webcast possible. Before we go, I would like to remind everyone that today's webcast will be available for on-demand viewing through August of 2018. You will receive an email from Labroots alerting you when this webcast is available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.